Hello everyone, welcome back to Swati's Bioscience Hub. So in the 3 sessions, we have been discussing about the cellular respiration in aerobic conditions. Okay, we have already discussed the glycolysis, the pyruvate dehydrogenase complex and the Krebs cycle. Okay, and the main motto of cellular respiration is to produce energy in the form of ATP. That means the main motto is ATP synthesis. Okay, and the ATP synthesis in cellular respiration takes place in oxidative phosphorylation, in this oxidative phosphorylation. So this oxidative phosphorylation, it takes place in the mitochondria. It takes place in the mitochondria of the cell. That means the ATP synthesis takes place in the mitochondria. So when ATP synthesis takes place, number of ATP produced and it is present in the mitochondria. That's why mitochondria is known as the powerhouse of the cell. Mitochondria is known as the powerhouse of the cell. Okay. So oxidative phosphorylation actually takes place in 2 ways. Okay, in 2 steps. It takes place in 2 steps. First one is electron transport chain and second one is chemiosmosis. It is chemiosmosis. So in 2 steps this oxidative phosphorylation takes place. That takes place in mitochondria. That's why mitochondria is known as powerhouse of cell. Okay, so here there are 2 steps. One is electron transport chain, another one is chemiosmosis. Okay, now as I said, it takes place in mitochondria. If we look at the structure of mitochondria is a double membranous structure. Okay, if you look at the structure of mitochondria, it is the outer membrane and these are the inner membrane which have some crystals, and inside crystals there is matrices. Okay, so this is outer membrane, this is the outer membrane and this is the inner membrane. Okay, and inside the inner membrane this is matrix. This is matrix, and the space between outer membrane and inner membrane is known as intermembranous space. Intermembrane space, that will denote as IMS. Will denote as IMS. Okay. So this inner membrane of mitochondria, it consists of a number of proteins. Okay, it has a number of protein in it. Okay, and all those protein complexes helps in ATP synthesis. Okay, so in glycolysis and Krebs cycle, we have discussed that in glycolysis and Krebs cycle, along with ATP and some other byproduct, we also get NADH and FADH2. Okay, that we have already discussed in our previous videos. I will give the link of those videos in the description box. Please check it out for the references. Okay, so NADH and FADH are the products of glycolysis and Krebs cycle. So here this NADH and FADH are used for ATP synthesis. These are the energy molecules. Okay, these are the energy molecules which will further be used to synthesize the ATP from it. Okay. Now, as I said, this oxidative phosphorylation consists of 2 steps. First one is ETC. So this NADH and FADH2, they will convert to ATP in both these steps. First ETC, then chemiosmosis. So first we will discuss about the ETC, electron transport chain. Okay. Electron transport chain consists of 4 complexes. It consists of 4 complexes which are known as complex 1, complex 2, complex 3 and complex 4. Okay. This is consist of 4 complexes. Complexes 1, complex 2, complex 3 and complex 4. So let's begin with the complex 1. So this electron transport chain that takes place in mitochondria, and these complexes are present in the inner membrane of mitochondria. So we'll start with the complex 1. We will start with the complex 1. Okay, let's say this is the inner membrane. Okay, this is the inner membrane of the mitochondria, and complex 1 is present here in the inner membrane of mitochondria. And the outside of this inner membrane there is IMS, intermembrane space, and here this is matrix. This is present between the matrix and inner membrane space, no? That. So what it does, as I said, NADH and FADH2 will be used for ATP synthesis. So in complex 1, here they will use NADH, okay, to produce NAD. It is complex 1 is also known as NADH dehydrogenase. NADH dehydrogenase. Okay. As the name suggests, dehydrogenase, then that means dehydrogenation process takes place. So what it will do, here this NADH dehydrogenase is present in the complex 1. So what it will do, it will remove the proton from NADH, and the NADH will convert to NAD. Okay, and then when it will remove the hydrogen molecule or the protons. So when it will remove the proton, along with it 2 electrons will also remove. Okay, 2 electrons will remove. Then this electron will pass to FMN, flavin mononucleotide. The electron will go to FMN or flavin mononucleotide. Then again this flavin mononucleotide will pass the electron to 8 iron sulfur complex. 8 iron sulfur complex. Okay. Now again this iron sulfur complex will pass it to the ubiquinone, which is also known as coenzyme Q. Okay. It will pass the electron to the ubiquinone. Okay. And ubiquinone is present outside the complex but in the inner membrane only. Okay. So it will pass the electron to ubiquinone. Now what will this ubiquinone do? It will accept 2 protons directly from the matrix. It will accept 2 protons directly from the matrix, and it will be reduced to ubiquinol. That is coenzyme QH2, that is ubiquinol. It will be reduced to ubiquinol. When ubiquinol is getting reduced in this complex 1, that's why it is also known as ubiquinone reductase. Ubiquinone reductase. Okay, this reaction complex 1 is also known as ubiquinone reductase. So while all this process going on, NADH is coming, NADH dehydrogenase is removing the proton from it, along with it is also removing some 2 electrons. Electron is transferring from NADH to FMN to into APA center, then to ubiquinone 2, and reduce it to the ubiquinone. When all this process happens, here 4 protons from matrix transports to intermembrane space. Okay, there 4 protons will transport from matrix to intermembrane space in the complex 1. Okay, this is the whole story of complex 1. Got it? Next, we'll move to the complex 2. Okay, as I said, 4 complexes. Now this is the whole story of complex 1. It actually reduces NADH dehydrogenase. Next, we'll move to the complex 2. Complex 2 is also known as succinate dehydrogenase. Dehydrogenation of succinate takes place there. So complex 2 is known as succinate dehydrogenase. Complex 2 is known as succinate dehydrogenase. Do you remember the Krebs cycle? If you do remember the Krebs cycle, then in Krebs cycle we have discussed in the 6th step there is a product succinate which was removing in the 6th step of Krebs cycle, removing hydrogen in the form of FADH2 and succinated converting to fumarate. Okay, this FADH2 is getting reduced in the complex 2 of the oxidative phosphorylation. That's why it is known as succinate dehydrogenase. Okay, now again we will discuss just like the complex 1, that this is the inner membrane and this is the complex 2. Okay, this is the intermembrane space and this is matrix. But complex 1 accepts the NADH, but complex 2 here accepts the FADH. It accepts the FADH2 and convert it to FAD. Reduce it to FAD. So while removing that hydrogen from FADH2, it also removes 2 electrons from it. It removes 2 electrons from it. This electron will pass to iron sulfur center of complex 2. Okay. Then again this iron sulfur will pass that 2 electron to the ubiquinone present outside the complex but inside the inner membrane. Okay. And here again this ubiquinone will accept 2 hydrogen or 2 protons. It will accept 2 protons directly from the matrix and here it will reduce to ubiquinol. Okay, it will reduce to ubiquinol. Okay, so this is the process that takes place in complex 2. But unlike the complex 1, there is no proton transportation from matrix to intermembrane space. In complex 1, there are 4 protons which are transported. Okay, 4 protons are transported. But in complex 2, no protons are transported from matrix to intermembrane space. Okay, there is no transportation of protons. It just reduces FADH2 to FAD. Okay, this is the story of complex 2. Now we have discussed the complex 2. Till now I have been keep saying that there is a ubiquinone is present which is accepting the electron from the complex 1. So now what is this ubiquinone? What is the ubiquinone? So ubiquinone is a electron carrier. Okay, it acts as a electron carrier which carries electron from complex 1 and complex 2. It carried electron from complex 1, now it is carrying electron from complex 2. Okay, and when ubiquinone is also known as coenzyme Q. Okay, so when ubiquinone accepts 1 electron, it becomes semiquinone. Okay, it becomes semiquinone. But when it accepts 2 electrons, okay, it accepts 2 electrons, it becomes QH2. That is ubiquinol. It will become ubiquinol. Okay, so this is ubiquinol and that gets reduced to ubiquinol. Okay, now understood what is ubiquinol and ubiquinol. And ubiquinol is the electron carrier of complex 1 and complex 2. Okay. Now, let's see the complex 3. We have already discussed the complex 1 and complex 2. Complex 1 is responsible for the reduction of NADH. Complex 2 is doing the reduction of FADH2. Now, complex 3. In complex 3, this ubiquinol carries the electron from complex 1 and complex 2 and it passes this to the complex 3. Okay. Now we'll see the complex 3. In complex 3, complex 3 is known as cytochrome BC1 complex. Why? That we will see. So let's, okay. So complex 3 consists of cytochrome B, then a risky center that is consist of 2 iron sulfur complex and a cytochrome C1. Okay, complex 3 consists of cytochrome B, risky center and cytochrome C1. As it contains cytochrome B and cytochrome C1, it is known as cytochrome BC1 complex. Complex 3 is known as cytochrome BC1 complex. Okay. Now what happens? It is also known as cytochrome reductase. It is also known as cytochrome reductase. Cytochrome reductase. From the name, you just can guess that there will be reduction of cytochrome molecules. That will be reduction of the cytochromes. Okay. Now we'll see the complex of the complex 3, the structure of complex 3 and the mechanism of complex 3. So this is the inner membrane again, and this is complex 3. Okay. As I said, it has cytochrome B, then it has a risky center, and it has cytochrome C1. Okay. Now, ubiquinol accept the electrons from complex 1 and complex 2. Then it gives it to the complex 3. Okay. So there are 2 ubiquinols. Okay. So what will happen here? These ubiquinols will give electron to the cytochrome B. So when it will give the electron to complex 3 accept. The first cytochrome B will accept that electron from ubiquinol. Then it passes this to the risky center. Then again it passes pass the electron to the cytochrome C1. And here cytochrome C1 will pass the electron to cytochrome C that is present in the inner membrane, but towards the intermembrane space. It is towards the intermembrane space. Okay, and it is present outside the complex 3. This is the matrix and this is the intermembrane space. So this cytochrome C will act as an electron carrier. Okay, and what it will do, the problem with cytochrome C is it can accept only one electron at a time. So here ubiquinol is bringing electrons, multiple electrons from complex 1 and complex 2, but cytochrome can accept only one electron at a time. That's why they use a specific cycle, that is Q cycle, that goes on inside it. Okay, they use a Q cycle where they complete the whole process in 2 steps. Step 1 and step 2. Okay, they complete the whole process in 2 steps. Step 1 and step 2. Now we'll discuss what is the step 1 and step 2, how the whole complex 3 mechanism takes place. So, first we will in step 1, the ubiquinol from NADH that performs this ATP synthesis process. Okay, so let's see, this is the inner membrane space. So this is the inner membrane. Okay, and and here we'll see the first step, and here we'll see the second step. Okay, this is the first step and this is second step. So this is intermembrane space, this is matrix, and this is step 1, this is step 1, this is step 2, okay, of complex 3. Okay, so now what will happen, the ubiquinol which of complex 1, first they will use the ubiquinol of complex 1. It will give the electron, it will reduce and produce 2 electrons. Okay, here it will reduce and it provide that protons, it removes 2 protons to the intermembrane space. Okay, it reduce and provide 2 protons to the intermembrane space, and also it generates 2 electrons. So now these 2 electrons, one electron will go to cytochrome B. Okay, from cytochrome B it will go to the ubiquinol present here or coenzyme Q. From cytochrome B it will go to the ubiquinol present inside the complex. Then when accepting this electron, when quinone accept one electron, it will become semiquinone. Okay, it will become semiquinone. And another electron will go to cytochrome C1. So it will go to risk center first. It will first go to risk center, then to cytochrome C1. And from cytochrome C1 it will go to cytochrome C. Okay, cytochrome C is a electron carrier here, so it will go to cytochrome C. The electron moves to cytochrome C. Okay, now in the second step what happens, here it uses the complex 1. Okay, it uses the ubiquinol from complex 1 or NADH part. Okay, NADH reductase part, NADH dehydrogenase part. Now here it will use the ubiquinol from the FADH2 or complex 2. So ubiquinol again comes and remove 2 hydrogens, 2 protons to intermembrane space and produce 2 electrons. One electron will go to cytochrome B. Okay, from cytochrome B it will go to the quinone. Okay, ubiquinol, and from ubiquinol here it will produce ubiquinol. Okay, here it will produce ubiquinol, and this then another electron will go to the risk center. It will go to the risk center, then to cytochrome C1. It will move to cytochrome C1. Okay, and from the cytochrome C1 it will go to the cytochrome C. It will go to the cytochrome C. So here cytochrome C is also an electron carrier. So there are 2 cytochrome C. So first it is from NADH, second ubiquinol is from FADH2. This is from complex 2, this is from complex 1. Okay, and now they transported the electron to cytochrome C. This is another cytochrome C, this is another cytochrome C. They are not same cytochrome C. So here they transported one electron to cytochrome C in first step. In second step it transported electron to another cytochrome C. Now we can see in the first step it removes 2 protons to the intermembrane space, and in second step it removes 2 protons to the intermembrane space. That means in total it is generating 4 protons and transporting into the intermembrane space. Okay, complex 3 is transporting 4 protons. So this is the story of complex 3. Now this cytochrome C will move this electron, will carry this electron to the complex 4. Okay, now we'll see the complex 4 of electron transport chain. So the complex 4 Here we will see the complex 4 of electron transport chain. In complex 4, this is the inner membrane. This is the complex 4. This is matrix. And this is intermembrane space. Okay. And here what happens, the cytochrome C that carried that electrons from complex 3, here this cytochrome C that will bring the electrons, and first it transferred to the prosthetic group copper A. Okay, then from copper A the electron will transfer to cytochrome A. Okay, again cytochrome will transfer that electron to copper B. Further this will transfer that to cytochrome A3. Okay, and here this cytochrome A3 will transfer that electron to oxygen. Here there is an oxygen ion is present which will accept the electron from cytochrome A3. So as it accepts the final electron from the cytochrome A3, it is known as final electron acceptor. Okay, here this is known as final electron acceptor. That's why this process is known as cytochrome C oxidase. Cytochrome C oxidase. And here this oxygen is the final electron acceptor. Okay, this is the complex 4. And in this process also matrix transport two number of protons to the intermembrane space. Okay, so this is the complex 4. Now what happens in electron transport chain, all these complexes made removed or transported electrons to the intermembrane space. Okay, transported the protons to the intermembrane space. So in intermembrane space there is a lot of protons are present now. Okay, that is known as proton gradient. In intermembrane space, all these protons which are removed from the complexes are stored now. Okay, number of protons, that is known as proton gradient, and this proton gradient will help in ATP synthesis. This proton gradient will help in ATP synthesis. At this point, at complex 4, the ETC, electron transport chain, is finished. Okay, now the chemiosmosis will start. The first part of oxidative phosphorylation is finished. Now the chemiosmosis will start. In chemiosmosis, the proton gradient which is present in the intermembrane space, that will synthesize the ATP. Okay, and there is a complex which is again present in the inner membrane, that will help in the transportation, help in the conversion. Okay, there is a complex 5 is there. Complex 5. Okay, so in complex 5, again this is inner membrane. It is a complex 5. There is a F0 is present, and the structure is having an F1 structure. Okay, that is at the tailbone point of the towards the matrix of the cell of the mitochondria. It has F1. So here what happens, four protons from intermembrane space, they undergoes phosphorylation and it converts one ADP, it performs phosphorylation, it converts one ADP or inorganic phosphate to it and produces ATP. So in complex 5, in chemiosmosis, we can see four protons are used to make one ATP. Okay, so if you think in the whole process, we have seen In electron transport chain there are 4 complexes. Okay. In electron transport chain there is 4 complexes, complex 1, complex 2, 3 and 4. So in complex 1, NADH convert to NAD, that's it is NAD dehydrogenase, NAD is dehydrogenase, and here it transported 4 protons to the intermembrane space. Okay. In complex 2 there is no transportation of protons. Okay, there is no transportation of protons, and here it reduced the FADH2 to FAD. It reduced the FADH2 to FAD. In complex 3, in both the steps, including both the steps, they transport 4 protons. In step 1 they transfer 2 protons, in step 2 they transfer 2 protons. In total 4 protons to intermembrane space. And in complex 4 they transfers 2 protons to intermembrane space. Okay, in complex 1 they transfer 4, in complex 2 they transfer 0, in complex 3 they transfer 4, in complex 4 they transfer 2. Okay, that means for one NADH and one FADH, okay, for one NADH is taking place in complex 1. So for one NADH they are transferring 4 protons plus 4 protons from complex 3, for 2 plus 2 protons from complex 4. That means total 10 protons. For one NADH they are transferring 4 protons to the intermembrane space. So when there is 10 protons, 4 protons is making 1 ATP. So one NADH will make 10 by 4, almost approximately 2.5 ATP. Okay, 4 protons is making 1 ATP, one NADH is making 10 protons, that means it will make approximately 10 by 4, 2.5 ATP. In one FADH2 is sending 6 protons. How? Because complex 2 does not transport any protons. That means it will only transport complex 3 and complex 2. That means total 6 protons. Okay, and 4 protons is making 1 ATP. That means here it will make 6 by 4 means 1.5 ATP approximate value. Okay, one NADH is making 2.5 ATP, one FADH2 is making 1.5 ATPs. Directly glycolysis and Krebs cycle also makes some ATP, and then makes NADH and FADH2. Later on this NADH and FADH2 moves to the electron transport chain and chemiosmosis, togetherly called oxidative phosphorylation. And here in this oxidative phosphorylation, this NADH and FADH2 will convert to ATPs. One NADH will convert to 2.5 ATPs and one FADH2 will convert to 1.5 ATP. So this is about today's discussion, oxidative phosphorylation. In oxidative phosphorylation we have discussed electron transport chain, and we have discussed the chemiosmosis. Electron transport chain consists of 4 complexes, complex 1, 2, 3 and 4. We have discussed all these complexes briefly, and we have discussed the complex 5 of chemiosmosis, and also we have discussed how this one NADH and one FADH is converting to the ATPs. So if you like this video, then put a comment in the comment box, like, share and subscribe my channel. Thank you so much.